This is podcast 12, and we've been walking through the different types of filaments in the cytoskeleton, and now we're on to microfilaments. These are the smallest diameter filaments. They are made of actin, units of the protein actin, and they're responsible for cell movement, streaming, amoeboid movement, telophase movement. This is not all cell movement. Some cell movement is due to microtubules, but a lot of cell movement is due to actin. And a couple videos to show you of that movement here. So here are movements of cells due to actin. You can see movement of this amoeboid cell here, the ruffled edge of a macrophage here. Let me go back on that one. Different cells and them moving, and that's due to actin. And here's another one. So when cells divide, you can see cells divide here, and you can see actin labeled bright yellow as a line down the middle, bright green, sorry, and it forms a contractile ring to split those two cells. So contractile ring down the middle, split the two cells in telophase. Okay. So in a thin filament, you have G actin subunits. These are, G stands for globular actin. It's a general name for a shape of a protein, a globular protein. So G actin is globular actin monomers. They polymerize into a filament. That filament has polarity to it, just like a microtubule. It has a plus end and a minus end. And just like a microtubule, the plus end has a net addition. The minus end has a net subtraction from it. But just like microtubules, doesn't mean that you can't add to the minus end. It's just that much more likely to add to the plus end. The plus end has ATP bound to the G-actin subunit, and soon after it polymerizes into a filament that's hydrolyzed to ADP, and then you have this thin filament. And it's not hollow in the middle, unlike a microtubule. It's pretty much solid actin. There is an assay called the barbed decorating filament assay, and I want to stress that this is not something that happens inside cells. This is just a preparation you would do in the lab to figure out what is happening in terms of orientation. So later on we'll learn about another motor molecule called myosin, and myosin binds to actin. Myosin has a head end that has the motor part of it, and then a tail end. If you just take a preparation of a bunch of those myosin heads and you throw it on an actin filament from a preparation, they will bind to that preparation in an arrowhead sort of formation. So you can just sort of see here, see these arrowheads, okay, like that, okay. And on those arrowheads, the minus end is the pointy tip of the arrow. So this is a mistake in the book. This should be minus. Okay, the pointed end is the minus end, and the plus end is the barbed end with the barb part of an arrow. So this is minus, and this is plus. So how you can remember that is if you have an arrow coming at you and this is the end you see, that's the minus, that's a negative thing for you. If that's the end of the C that you see, that means the arrow is going away from you, that's a positive thing. So once again, this is not something that happens inside the cell. This is an artificial condition, just an assay to find out what is the orientation of these filaments. And you use a whole bunch of myosin heads to figure out where's the minus end and where's the plus end. And so this end here would be the end that was growing, and this end here would be the end that it started on, or the end that was shrinking. There are proteins associated with actin filaments, just like there's proteins associated with microtubules. They have different functions. You have proteins that help polymerize the actin filament, make it grow. Those are an example of that would be formin. You have filaments that can sever proteins that can sever actin filaments if you're remaking the shape of a cell. So the cell is going through some sort of transformation. And that would be an example. That would be gelsolin. You have filaments that cap the end of an actin filament in cells where you want your actin cytoskeletal array to be really stable, like in muscle, for example. You don't want to polymerize and depolymerize your muscle all the time. Cap Z is an example of those kinds of proteins. And then you have filaments that cross-link different actin filaments to create the cytoskeletal network. And those examples of those would be filamin and alpha-actinin and fibrin. In the last podcast, we talked about cilia and ciliated cells. Those have tubulin inside of the cilia. In microvilli, there's actin inside the cilia, so not the same thing. So this would be like what are the cells that are lining your gut, versus the ciliated cells would be like cells lining your throat. But in microvilli, where you have these projections that increase surface area, these are supported by actin network of actin filaments, and they are all of the same orientation, plus end on one end, minus end on another end. And they have several cross-linking proteins, so that this is a very stable structure. You can also have an inner network of actin underneath the surface of a membrane, to which you can have membrane-bound proteins anchored. So we talked about this when we talked about the fluid mosaic membrane, how some proteins are anchored in the fluid mosaic membrane, sort of like a buoy is anchored to the bottom of the ocean, and the ocean travels around the buoy. So the bottom of the ocean in this case is an actin network of cytoskeleton, and these proteins attach to that, and then the fluid mosaic membrane moves around those proteins. And these type of proteins are like band 3 in red blood cells, for example. This is an electron micrograph of that sort of network, where you have, you can see the actin network connecting these buoyed proteins in a membrane. One spectacular version of how actin polymerization can be used to power movement is with the bacterium Listeria. And in pregnant moms who could be infected with Listeria, every once in a while you hear about a Listeria outbreak. These bacterial cells can polymerize actin dramatically on one end of the bacterium and polymerize it so that it moves the bacterium. And I'll show you a movie of this in a second. And the bacteria will rocket all around the cell, even rocket into the plasma membrane, so much so that a neighboring cell may take up that projection of a Listeria rocket, and it'll infect the next cell. So this is pretty dramatic. I'm going to show you this movie here.
move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake.
So the black things are the Listeria bacterium inside the cell.
Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path. If we look closely, we can see a bacterium divide inside the host cell. Immediately after separation, the two daughter cells assemble their own actin tails and start moving about. These bacteria can also form actin comet tails and move in cell extracts. Here, the bacteria are expressing the green fluorescent protein, and actin is labeled red with a fluorescent dye. The dynamics of the actin tails that propel the bacteria through the cytosol can be modeled based on known biochemical and physical properties of actin and actin filaments.
So it's not that the actin is wagging back and forth to cause this thing to propel. It's actually pushing the bacterium ahead of it as it polymerizes. It polymerizes so fast that it's pushing the bacterium ahead of it. And these bacteria have evolved this mechanism to move within cells and actually move from one cell to another cell. Pretty cool. Okay. And an H5P question for you.